Good evening, centers. Shillong. I can see Shillong. Good evening. How are you? Yes, children. How are you? And how was your paper on Saturday? Most important of all. Yes, children. Are we connected? Yes, Shillong, I can see. Yes, children, tell me. How was your paper? Students are there at Shillong Center? Or are they yet to come? It was okay? It was easy or difficult? How was it? Yes. Was it easy? Yes. Okay, fine. Neutral. Nice. You must be getting good marks after all. Okay. So, okay, any doubts on the paper? Do you have any doubts from that paper that was done on Saturday, that was asked, you were solving on Saturday? Any doubts from that? Yes, children? Yes. Do you have any doubts? Any doubts? No? No doubts? Wish to ask something? Yes. No? No questions? Okay, fine. Yes, Shillong? Tell me any questions? Yes, children. There are many children sitting at Shillong. Any questions from the paper? Yes, you can ask me. It was an objective paper. This must be first time in your whole academic career. You must be... Okay, fine. Okay. But no worries, you will be having all the chapters, what we are running with. That is body fluids and circulation. When you are going to give the test for these chapters, you are not going to face these problems, isn't it? So we will be having backup of all those classes later onwards. But yes, we will be doing these particular chapters in a very, very good note, on a very, very good note, in a way that you are able to solve these kind of questions. Yes, along with that, we'll be discussing some of the important questions that is important from your school exam also. That is going to be asked in your school exams also. So I will be having like two modes. One will be for NEET and one will be for your school exam, because we cannot ignore either of them, isn't it? But obviously, that should be from your part as well, that you are ready to answer the questions when I am giving you on subjective content. So if I am asking you questions on subjective part, you should be ready to answer those questions as well. So if I am planning a test for, say, 1 marks, 2 marks, you should be able to do that test honestly, and obviously, how can we judge it? Obviously, you need to give me a screenshot or a kind of photo of those papers in a way that we can improvise it. Marking will not be obviously the priority. It will be improvisation, the content of your answers. Okay? Fine. So, what were we talking about in the last class? What was our topic of discussion in the last class? Tell me. What was we were talking about in the last class? I'm not able to see many of you in your class. Is it like we need to have some lights? Shillong? I'm not able to see. I'm just able to make out there are many heads in the class. But I'm not able to see clearly. So can you just make sure we can see each other? Yes. If you're giving me a thumbs up, I won't be able to see you. Yes. Okay, fine. So somewhat clarity is there. Okay. Okay, fine. So what were we talking about in the last class? What was the topic of discussion? I guess we're talking about blood, isn't it? We thought of like blood, we thought of differentiating blood into two different components, that is plasma, first component, and the formed elements. Remember? Just have a brief review. So plasma was the watery part, it was yellowish in color, it contained many different kinds of things. Remember we discussed osmosis also, that how albumin helps you in osmosis. Then we talked about these particular things in the last class, and we talked about formed elements also. So we thought of three formed elements. The first was RBC, the second was WBC, and third was platelets. So RBC is what? RBC are the red blood cells. We got a full explanation about RBC. I thought of, we talked about much in detail about RBC. I told you some extra things also about RBC that is not in your course syllabus. But yes, we tried to discuss as many as things possible. Then we thought of WBC. WBC was also well explored. WBC again, we did two different types of WBC. The first was the granulocytes and second was the agranulocyte. So granulocyte was the one which had granules like neutrophils, basophils, eosinophils, and the agranulocytes were not having any granules, and they were said to be what? They were said to be monocytes and lymphocytes. Now we are going to discuss today the third formed element, that is the platelets. Let's see. So this is body fluids and circulation. So we are going to have platelets. So what are platelets? Platelets are the third type of formed elements, isn't it? So what we did in the last class, just an outline. We have RBC, we have WBC, we have platelets, isn't it? We have three different types of cells. So now, when you talk about platelets, what thing comes into your mind? What do you think? What should be the platelets? Yes? This again are the typical cells which do not have nucleus, okay? The number varies between 1.5 lakh to 3 lakh in single mm³ of blood. So they don't have nucleus. Why? Why they don't have nucleus? They are formed because of a special cell called as megakaryocyte. So what is the story like? We'll have a quick look at the story. This is a particular cell with a big nucleus. And what do you call this cell? You would call this cell to be mega. Mega means large, karyo means nucleus, cyte means cell. So obviously it is a particular cell with a big nucleus. Now what happens is, this cell undergoes fragmentation. So we all know what is fragmentation. It starts to break down into many specific parts, many specific pieces. So this is what is the fragmentation kind of thing. So when they fragment, now they are called as platelets. That is why we said in the initial point only that they don't have any nucleus. Why? Because they are formed by fragmentation of special cells. And what are the name of the cells? The cells are said to be megakaryocyte. So what is platelet doing? Basically, what is platelet? The function, like you studied, RBC was doing what? RBC was helping in transport of oxygen, isn't it? We did that thing. We have done respiration and breathing exchange of gases in much detail. We all know what is RBC, how is RBC important for transporting both oxygen as well as carbon dioxide. Now, when you talk about WBC, we know WBC is a part of your immunity, is a part of your defense system, isn't it? We did that all those things in last class. And when we talk about platelets, what we talk about is basically the particular component of your body that deals with blood coagulation or blood clotting. So your platelet is playing a very, very important role in your body. It is helping in clotting of the blood. Whenever there is a trauma means an injury, what happens? There is a huge loss of blood. There is a huge loss of blood from our body, but then comes the role of the platelets. How? Whenever there is trauma means injury, what does trauma do? Obviously, a trauma is going to damage the platelets also. So platelets, they get damaged because of trauma. Trauma means any kind of injury, any kind of problem where there is not only bleeding, but yes, bleeding can be internal as well as external, both. So platelets get damaged. These damaged platelets, they release a chemical called as thrombokinase. And this thrombokinase, what does it do? It activates the prothrombin. So prothrombin is what I have always taught you these things, that when you talk about pro shows an inactive stage. Pro, gen, this is what? These are inactive stage. Like we studied in digestion and absorption, pepsinogen. We talked about pro-renin, isn't it? Then these particular kind of things are what? Whenever you put a pro or gen, prefix pro or suffix gen, gen, you talk about inactive state. So in inactive form, prothrombin is always present in our circulation. It needs to be activated by an enzyme, by a chemical called as thrombokinase. And the source of thrombokinase is platelets. Yes, good evening, Tamil Long. So source of thrombokinase is what? Damaged platelets. So until and unless the platelets are damaged, we are not going to have any thrombokinase. So this thrombokinase is going to convert your prothrombin into what? Into thrombin. So your thrombin is going to be converted. You get what you get at the end of the day, you get is prothrombin. Sorry, a thrombin. Now this thrombin is very, very important. Let's see why. This thrombin is again going to start another set of reactions where, with the help of calcium ions, it is going to convert fibrinogen into fibrin. So with the help of, we're talking about platelets, we are continuing with the same topic that we were talking about in the last class, that is body fluids and circulation. And this is, we are just undergoing the platelet section. How is platelet going to help you out? So this is what is the topic of discussion. And platelet is going to do what? It is going to clot your blood. It is going to prevent the excessive loss of blood from your system, from your circulatory system. So what is fibrinogen? If we talk about fibrinogen, what comes into my mind is, it is soluble factor. And what is fibrin? Fibrin is insoluble. Moreover, if it is insoluble, this must be having a particular structure also. Yes, it has a thread-like structure. So what happens is, this fibrin, they start to form the thread-like structure and they form a very fine meshwork like this. So this meshwork is basically used to prevent or to block the formed elements. So all the formed elements would be now behind this meshwork. Suppose this is my RBC. RBC is blocked. Suppose this is WBC, WCBC is blocked. So what is this meshwork doing? This meshwork is actually preventing the formed elements to get lost from the body, to get gone from the body. So thread-like structure, what they do? Form network. And prevent the blood loss. So this is a basic process, this is a basic pathway what platelets initiate exactly when they get damaged, and the end of the whole story is what? We get the blood clotting. Okay? Is it clear? Shillong, Tamilong? Are you clear up to this? Just give me a thumbs up if you are clear. Yes, children, clear? Yes, Tamilong, very good. Nice. So this is what is once again platelets, formed elements, the last part, no nucleus, just like RBC. So now you have two formed elements without nucleus. What are they? They are RBC and platelets. So what does platelet do? Platelets help in blood clotting. How? This was the pathway I was talking about. Trauma, blood platelets get damaged, release thrombokinase. Thrombokinase converts prothrombin into thrombin, fibrinogen into fibrin. And then this fibrin is what? These are the thread-like structures which are actually preventing the blood loss. So they prevent the blood loss from the system. Okay. So how many of us have observed that after blood loss there is a watery fluid coming out? How many of us have observed? Am I the only one to observe it? How many of you have observed this thing? That when you have a wound and after the wound is clot, after some time there is a liquid white, transparent fluid coming out of the wound. How many of you have observed this thing? Yes. That's me questions. Have you? Just raise your hand, Shillong. Tamilong can just tell me. Shillong, just raise your hands if you have observed. Yes, I have also observed. Okay, fine. So many of us have observed. What is that watery fluid? So blood is what? Blood is plasma plus formed elements. Okay? What is plasma? It is blood minus formed elements. Then we have a serum. That watery fluid is called as serum, that oozes out of the wound after the clotting process has been done. Why? Because serum is the particular fluid from plasma minus clotting factors. So you have a set of clotting factors like you have seen over here. This is a clotting factor. This is a clotting factor. This is also a clotting factor. This is also a clotting factor. This is also a clotting factor. So when these particular clotting factors, they are used in a reaction, okay, they are now over. They have been used up. Nothing, no clotting factor is left. What happens is the plasma without the clotting factor forms what? Forms serum. And this serum is the particular fluid that oozes out of the wound after the clotting process. So this is important. All these equations are very, very important. So what is serum? What is serum? Fluid that oozes out of the wound once clotting process is over. Why? Because serum is plasma minus the clotting factor. Just let me know if you have copied. So when clotting factors are used up, we are left with what? We are left with serum. Okay. Clear? Tamenlong, Shillong? Very good. Okay, done? Clear, everybody? Written, copied? Tell me, children, copied? Yes, children? Yes, Tamenlong, Shillong, done? Done, just give me a thumbs up, Shillong. Tamenlong is like they have done. Shillong, just give me a thumbs up. Can be. This can be called as defibrinated blood, but yes, this term is not that frequently used. We use serum on a frequent basis. Okay. Can be called defibrinated blood, but yes, defibrinated blood will be a very odd term, because see, we are not only removing fibrinogen, but we are removing many other components also. So there are in total, I just wrote over here just like how many components I had marked. 1, 2, 3, 4, 1, 2, 3, 4, 5. Just marked out 5, but there are like 13 clotting factors. So we cannot write for every clotting factor. This is decalcinated blood. This is dethrombokinetic blood. What we do is that we have a particular term called as serum. And this is not blood actually, because plasma is a part of blood, not the whole blood as a whole. Okay, so you cannot take plasma to be a blood or to the whole blood as a whole. What you need to have is, you need to have as a component of plasma. This blood will be what? Blood will be the component of plasma. Clear? Am I clear, Shillong? Okay. Fine. So I'm continuing ahead. Okay. So what is your blood group? What are the blood groups? Let's see. What are the blood groups present in our class? Yes, Tamenlong, you start. Start to mention everybody's blood group. One of you please sit on the keyboard. Sorry, but one of you just sit on the keyboard, please, and just mention everybody's. Everybody cannot be O positive. Yes, we have a variety of blood groups. Just write down everybody's blood group fast. Yes, Tamenlong, you do the same thing. Just let me know what are the blood groups present at your center. Different types of blood groups, what do people have? Yes, well, there is one answer. There's an answer from Shillong, O positive. Wow, nice. Okay. I will tell you later why it is nice to have O positive. What is nice about, like, why I am happy for O positive. Okay. Yes. Tamenlong, please write the blood groups present at your center. All of you. Yes. Quickly, start writing. How many of you don't know your blood groups? Is there somebody among us? Is there somebody among us who does not know? Please raise your hands. Okay, we will use that person as an experiment in this particular class. I'll just run a small experiment in the class, and we will be giving them an opportunity to work in that experiment. Yes. Nobody? Shillong, all of you know your blood groups, but I have only two results from there. One is having A and O positive, and Tamenlong, they have mentioned nothing. Please, be fast. Children, quickly. I can see many heads at Shillong. Why only 2 blood groups? Tamenlong, please children, write your blood groups. If you know, obviously. And if you don't know, just let me know you don't know. Yes. Okay. Fine. So how many of you are O positive? Just raise your hands at Shillong Center. How many of you are O positive? O positive first. Yes. How many of you are O positive? Okay, fine. And A positive? How many of you are A positive? Okay. Fine. Tamilong? Children yet to answer. Okay, I am also A positive. I am also among one of you. I am also A positive. Okay, let's see what is this O positive, A positive, B positive, isn't it? B positive. And then we have some people AB positive. Some people are there who you will find them to be O negative, A negative, B negative, AB negative, isn't it? So there are basically different kinds of blood groups, and these all different kinds of blood groups, they are a part of, let's start blood grouping. They are all a part of the ABO blood grouping system. So what you have is, you have ABO blood grouping. But this ABO blood grouping was given by Karl Landsteiner. So this was given by Karl Landsteiner. And what was Karl Landsteiner doing? He mentioned different types of blood groups ABO. So how can you conclude that this is that particular blood group, this is that, all those things? Let's see. This is your RBC. Your RBC has an antigen and based on this antigen, you have a blood group. So if it is an A antigen, your blood group would be A. Okay. Again. This is the RBC. This is having B antigen. In that case, your blood group would be B. So this is B antigen. This is A antigen. If somebody's RBC has both the antigens, A as well as B, he or she will be having AB blood grouping. And then again, if there is no antigen, nothing, the person would be said to have 0. It's 0 or O blood group. Okay, clear? Now can I give the person with B blood group, can I transfuse him with A blood group? There is a person with B blood group. Can I transfuse him with blood from A blood group? Tell me. Can I? Yes, children. Can I transfer? Yes or no? Tell me. No? Why? You're right, but why? If I am A positive, can I take blood from somebody who is B positive? See, if I have blood group of A, that means I have anti-B antibodies in my blood. What I have is I have anti-B antibodies in my blood or in my serum if I am having blood group A. Now if somebody tries to transfuse me, transfuse means blood, obviously blood interchange. If somebody tries to give me blood group B, these particular antibodies that I have just now written, these particular antibodies, that is anti-B antibodies, since the name itself is anti-B, means they are against the B. That is, they are against the any RBC which has this antigen. So what is it going to do? It is going to start a particular reaction where there will be clumping of blood. The blood will be clumped and forms clot in the vessels. And because of that particular issue, the person can die also. Are you getting my point, everybody? So, if a person is having blood group A, his system will be naturally having anti-B antibodies. And if it is blood group B, they would be having anti-A antibodies. So, all those people who are having A blood group, me also included, they have anti-B antibodies. I have anti-B antibodies in my system. If somebody tries to transfuse blood in my system, which is blood group B, what is going to happen? The anti-B antibodies will start to react against this antigen. Once it starts to react, there will be some fighting, okay, action, and after action, what is going to happen? There will be some clot-like formation, and that clot is going to be very, very harmful if considered the flow of the blood. So the flow of blood is going to be compromised because of that clot formation. The person can die also. Similarly, if a person has AB blood group, his body will be having no antigens. Sorry, no antibodies. So you can understand this from a simple example that if you have seen somebody, if you have already met somebody earlier, you will not resist. Isn't it? You will not resist is like if I am your friend, for example, and if I come to your home, you will never say no to me. Why? Because you already know me. Similar is the case with AB blood group also. Now AB blood group, the person with AB blood group knows everything. I know what is A, I know what is B, so I will not be having any problem with any of the people coming to my place. If A comes to my place, most welcome because I know who is A. If B comes to my place, B is also welcome. But if I am only aware about A, and if I am encountering B, I don't know what is B. So what my body will be doing? My body will be fighting it back. If my blood group is B, I don't know what is A. So what my body is going to do? It is going to fight back. So it's just a basic response that whatever my body does not know, it is going to fight back that particular thing. It is going to act against that thing. Similarly, if somebody's blood group is O, O means zero antigen. Now his blood group is not aware about any of the antibodies. It neither knows A nor B. So it's very, very naive. It's very, very like what we say. Does not know anything. What is antibody? What is antigen? So what is O going to do? O will be treating A also as an outsider. A would be treating B also as an outsider. So it has two different kinds of antibodies in its blood. Anybody with O blood group, they have to make a special note of it that they have both the kinds of antibodies in their blood since you don't know about any antigens. O means zero. So you don't know about any antigens. What you're going to do? You're going to fight back with each antigen you are encountering with first time. Let me know if things are clear. Yes. Long story, but let me know if the things are clear. Shillong, Tamilong. So this seems to be complicated, yes. So this was Karl Landsteiner's part of work. We should be thankful to him. He made us clear about many different facts. Okay. Fine? Clear? Okay, nice. So now if I make another table where I am having B, A, B, O. Okay. So antibodies, you tell me. What are antibodies? Obviously there will be anti-B, it will be anti-A, nil, anti-A and anti-B, both the antibodies. Now what we have is, next, what do you think? What it should be? When you talk about antibodies after this, what do you think about it? Tell me. Next thing. Yes. Can donate blood to. Now you're going to answer me these questions. I'm not going to solve it. Can donate to. So it's useless studying everything if you're not concentrating that, yes, from whom I can take blood and from whom I cannot. Who can take my blood and who cannot. So we should know these things. This is what is a practical approach of blood group system. Okay, so can donate to. Tell me. To whom blood group A can donate blood to? Blood group A can donate blood to whom? Yes. So it can donate to A, first of all. Exactly. Can it donate to AB? Can it donate to B? First of all, tell me. Can it donate to B? Tamilong, Shillong? A can donate to A, yes, very much clear. Very good. B can donate to B. AB can donate to AB. O can donate to O. Done. Can receive from A. Can receive from B. Can receive from AB. Can receive from O. Done. Next thing. What about B? Can it receive from B? Can it receive from B? No, it cannot. Why? Because B has what? When B will be transfused into A's body, so I am A, if I am getting blood group B in my body, what I will be having? I will be actually starting a set of reaction that starts the antibody production of B, antibody B, anti-B antibodies. So these anti-B antibodies are going to do what? They are going to start a reaction where the blood cells will get damaged, clotting will happen, and after clotting the person can die also. So we will not have A going to give blood to B, neither it can receive from B. But yes, it can donate to AB. Can it? Yes, it can donate to AB. Why? Because AB has no antibodies. AB has no problem in receiving blood from either A or B, since there are no antibodies. So it can donate to two of them, that is A and AB. It cannot donate to O as well, because O has both the antibodies. Okay. Now B can donate to B and AB also. AB can donate to AB, and apart from AB, it cannot donate to B, not to A, not to O. Because if it donates to A, this B part, this section B, this is going to have a problem in the body. Why? Because the person does not know what is B. It knows what is A, but it will not know, the person's body does not know what is B, so there is a little problem. O can donate to O, since no antibodies, no problem. It can donate to A, sorry, no antigens, no problem. So O can donate to A, B, and AB as well. Clear? Can receive from A and only O. It has a problem with AB as well as with B. B can receive from B and O. AB can receive from A, B, and O. Since it has no antibody, so it can receive from everybody. O can receive from only O. Because it has no antigen and both the antibodies, it is going to be a problem. So from this chart, we can very well accept that this happens to be universal donor and this happens to be universal recipient. So donor can donate blood to everybody, that is O, and recipient can receive blood from anybody, that is universal recipient. AB happens to be universal recipient and O happens to be the universal donor. Clear? Let me know if you have any problem. Shillong, Tamilong. Are we clear up to this? Yes, I know it's a bit complicated, but yes, it is not that complicated too. It can be easily understood. We are all aware with these practical issues. We know these things on a practical basis also. Okay, fine. No issues? No issues? Shillong, Tamilong? Okay, very good. Now I talked about one antigen. Now I'm going to start the increasing. I'm going to start increasing the number of antigens. Okay. Now you have another system that's called as RH blood grouping. Now this RH is a particular term which is similar to rhesus monkey. This is a factor because O has both the antibodies, and if you are giving blood that is AB, isn't it? AB has both the antigens. See? O has both the antibodies and AB has both the antigens. So it's a clear-cut matter of conflict. If already there is somebody who is ready to fight, isn't it? Antibodies are ready to fight. And you are giving them the reason to fight. Antigens are the reason to fight. And antibodies are the ones who fight. So if you are giving them the reason to fight, they are going to fight. Isn't it? So antigen is something which is going to give reason to the body to fight against something. And antibodies are the fighters. Okay. So if you are giving something, this is like the particular element which will initiate the fight, obviously the fighters are going to fight against it. So here there are two reasons, not single, but two reasons. That is, first reason is A antigen, second reason is B antigen. So both the antigens are going to give a cause to anti-A antibodies to fight, anti-B antibodies to fight. So there will be a like World War III like condition going on. Okay, fine. Nice. Okay. So RH blood grouping system, this is RH factor. If you talk about this factor is basically named RH because it is similar to a factor which is found in rhesus monkey. Now what is this RH factor? Let's talk about. Now I told you that this is your RBC, isn't it? I told you a story like this. That this is your, I will make it in, you also make sure you are making this in the center of your page because I will be using the either sides of your page. This is your RBC. And what was the color in A antigen? This was green, this was magenta. Okay. Okay, fine. A antigen. Let's have a bright color. This is Rh antigen. What is this? This is antigen A and this is antigen Rh. Okay, now two cases can happen. Either Rh factor can be present. Not able to draw a complete. Two cases can happen. Either Rh factor can be present or Rh factor cannot be present. If Rh factor is present along with A antigen, my blood group would be what? My blood group would be A positive. If Rh antigen is absent but A antigen is present, my blood group would be A negative. So if Rh factor is present, plus NT means present, the person is said to be Rh positive. And if Rh factor is absent, minus NT for absent, the person is said to be Rh negative. Clear? So, I am having two different blood groups with antigen Rh, A positive, without antigen Rh, A negative, and that too in coordination with A. Next, again, this is my RBC. I have an antigen which is of this color. Excuse me. I have an RBC. I have a RBC with this antigen. Okay. So since I have many colors, I can make it very, very colorful, you see. Fine. So this is what? This is B and this is RH. So if it is B and RH together, it is B positive. B without RH is B negative. Clear? Okay. Let's explore some more. Now you can tell me, isn't it? If both the antigens, this is green, don't make both the antigens at the same place, have some space. This is again this color. All the antigens are present. A, B positive, RH, A antigen, B antigen, everything is present. Now, the last one, RBC with no antigens, nothing, and with just one antigen. What is the antigen? That is RH. Simply O positive. No antigens at all, nowhere, O negative. Clear RH system of blood grouping. This is what is called as RH system of blood grouping. Tamilong, Shillong, clear with this? Yeah, clear? Nice. Tamilong? Very good, Shillong. Yes, Tamilong, clear? Did you copy that? Okay. Clear? Just give me a thumbs up, everybody, Shillong Center. I can just look at everybody. Okay. Yes, children. Okay, very good. So now you can make an idea. You can have an idea. Tamil Long, what is taking so long? Any problem? Just let me know, please, children. I will be more than happy to sort it out. Yes, Tamil Long, let me know if any problem, please. Clear? Clear with Shillong? Now you can have an idea who can donate blood to whom with more clarity. Let's have it. We'll have a MCQ round. Yes. Good question, Shillong. I was about to ask this thing. Okay, fine. Nice. So if I am A positive, okay, I will be very selfish if I'm telling this thing. See, but nature has made me, so what can I do? If I am A positive and there is XYZ person who is A negative, okay. So if I am A positive and there is somebody who is A negative, I can always take blood from him or her, obviously, but I will never give blood to A negative because my body has an antigen which his or her body does not have, and that antigen is Rh antigen. So Rh antigen can also create a problem. That is, it also generates anti-Rh antibodies because my body, it has seen what is Rh. It has no problem with Rh or without Rh. But if there is somebody whose body has never encountered what is Rh, suppose this person who is A negative, he does not know what is Rh. Okay. So if they are given this particular Rh positive blood, what is going to happen? Their body is going to fight back. I told you, antigen is a reason to fight and antibodies are the fighters. So if I am giving them a reason to fight, that is what the reason is now, Rh antigen. So Rh antigen, when introduced in their body will respond with a fight back response, and that is going to be very, very fatal for them. Okay, so A positive cannot donate blood to A negative because of Rh antigen. So whatever antigen your body has not seen, they are all outsiders. For a person who is A negative, Rh factor is an outsider for his body. For a person who is O negative, all the factors, all the antigens, A antigen, B antigen, Rh antigen, all three of them are outsider for his body. So it totally depends on what kind of fighting things, what kind of reasons you are exposed to. If I am exposed to A antigen, I am exposed to A antigen in fact, because I started the class with the note that I am A positive. So I am exposed to both A antigen, I am exposed to Rh antigen as well. My body knows what is it. So if I am given something which is not having Rh, I don't have any problem. With or without, I don't have any problem. If I am getting a blood which is having Rh positive thing, that is A positive, I will not have a problem. Okay. But yes, I will have a problem if I am getting blood from A, B, positive, negative, anything, because B is something new to my body. I don't know what is B. My body has never encountered what is B. So it will react in a very different manner. Am I clear? Everybody? Tamil Long, Shillong? Okay, let's have some MCQs. Let's see how much clear we are. Okay. So, my first question is, a person met a road accident. You can select multiple options, like you can have A, B, C, you can have multiple options. Okay, you just need not select only a single option, but yes, you can select many options, because I will be in short writing all the options over here. E. Okay, fine. Complete. So you need not have only one option from here, but you can select multiple options. Yes. It's a person who met a road accident. Take your time, no issues, but please give correct answer. Found to have blood group AB, negative. And he can receive blood from AB negative, B positive, AB positive, B negative, A positive, A negative, O positive, O negative. Quick. Multiple answers. Tamilong, are we lost? Connection? Tell me, children. And since long you are not answering anything. It has been 10 minutes since your last response came. Yes, children, any problem? Just let me know if... Okay, we are here, but answer, please. Your answers are not here. Please answer the questions, children. It's a good question. This is like, basically, yes. Answer this question then. Answer, please. Yes, I have got a response from Shillong. Let me see. Just let me see. What is it about? Yes. Tamilong, answer this question, please. Today I'm not able to see many of my children. Kohima is not here. Itanagar is not here. Many of the children are not here. Okay, let me help you out, Tamilong. Just let me know, are you doing this question? Are you trying to do this question or you don't have any idea about it, children? Tell me. Are you doing this question? This is important, children. This is a very, very important question. These kinds of questions, they need to be solved to build you. Exactly. Take 2 minutes more. No issues. Take 2 minutes. Try. So we can do one thing. Just let me know what have you tried. Just start typing whatever you are like trying. We will discuss. Okay. Just let me know what have you tried from your end. Just give me A, B, C options like that. Because I have already written all those things. You just give me the numbers. See, this all story started with the fact that anybody who is Rh positive cannot donate to Rh negative person. But if somebody is Rh negative, he or she can donate to Rh positive. So you can just reverse the order and you can write one more theory that Rh negative cannot give to Rh positive and Rh positive can give to, Rh positive can take from Rh negative. So this person is somewhat having only two antigens, that is A and B, not having Rh. So obviously he can always take from somebody of his own client, isn't it? This is from his own client, so he can take blood from AB negative. Since he himself is AB negative, he's having similar kind of body composition. He or she cannot take from B positive because this is going to be problem. I just mark the things which are going to be problem. This is going to be problem in their body. They again cannot take from AB positive because this is going to be the problem in their body. Okay? Yes, but they can take from B negative because this person who has met the road accident, he or she has seen what is B. Although they don't know what is RH, but D also does not know what is RH. They can take from him. RH again is a problem. No RH, no problem, because this is somebody who has already A antigen. So he does not have any problem when A antigen is given to his body. This again is a problem. O negative, no antigens. No antigen means no problem, yes. So this can also donate. So finally the list is somewhat, he can take blood from AB negative, B negative, A negative and O negative. So these rest of the blood groups can be just eliminated out. So we have the person, we can allow the person to take blood from AB negative, from B negative. So is your answer somewhere near this Tamil long? Just try to understand this thing. If it is not, just let me know. If it is not clear, just let me know. So the pink ones can donate blood to this person. Or the pink ones, AB negative can take blood from these people. Okay. So if it is clear, okay, it's clear Tamil long. Okay, very good. So you can sort it out questions like this. Okay. Okay, so let's take a small break. Do you wish to take a break or shall I continue? It's your wish. Wish to take a break? Okay, Shillong. Fine. So we'll take a small break of 5 minutes. Just freshen you up yourself. Try to discuss who can donate blood to whom. Okay, and we'll come back after the break. Resume after the break. Yes, back from the break, children. Relaxed? Enough of blood grouping today, isn't it? Too much of hodgepodge things. Okay, Shillong. Back? Everybody's back in the class? Okay, Tamilong? Okay. So shall we start? Okay. Tell me, shall we start the next topic? Okay. So I'll just show you a small slide. Okay, I guess it's not here. Okay, fine. Anyhow, we are clear with all the things. So we are now up to date that yes, whenever you talk about blood circulatory system, whenever you talk about all these things, what is basic idea behind all these things, blood grouping and everything. So there's a particular case which is called as erythroblastosis foetalis. Okay? Am I frightening you with the name? Okay. It is called as erythroblastosis foetalis. What is it? Erythroblastosis foetalis. So what is unique over here? Erythro means red blood cells, blastosis means obviously bursting, blasting. Foetalis refers to of fetus. Yes, it's correct. You are listening correct. It's fetus. So let's see what is erythroblastosis foetalis. This is a very special case when a Rh positive male marries a Rh negative female. Okay, the male has to be Rh positive and the female has to be Rh negative. The reverse will not work. So if the male is Rh negative and female is Rh positive, this will not be the case. It has to be in a way that female is Rh negative and male is Rh positive. So what can happen? In the first pregnancy, chances of both Rh positive and Rh negative fetus are equal. So if a female who is Rh positive and carrying a fetus who is again Rh negative is of no, this is going to give no problem because they share the same blood group. That is, female is also Rh negative and the fetus is also Rh negative. So it's not going to be any problem. The problem can arise if the fetus is Rh positive. So first of all, what happens is the fetus is actually covered by a barrier. There's a barrier in between the maternal and the fetal blood, and the barrier is called as placenta. Okay. So Rh positive fetus, let's see what happens. This is something which is different, isn't it? This is positive. Look over here, everybody. This positive, this is negative. So this is something which is of different nature. So initially what happens is this placenta forms a barrier and prevents any mixing of blood in between fetus and mother. So we should be aware of this fact that mother never supplies direct blood in the fetus. But yes, the particular elements, the particular nutrients, components, they are finally selected by the placenta and sent to the fetus by the mechanism of diffusion. So it's not like the mother's blood is running into the fetus. Never. Fetus prepares its own blood. It has its own system. Mother's blood never enters into fetus. But yes, the components required to make RBC will be supplied by mother. So the raw materials will be from the source of mother. Whatever mother is eating, the child is going to get those nutrients like folic acid, iron, calcium. The child will be getting that from mother. But that all will be in raw material form. So it's actually fetus who is going to make its own system, because many times you know that, like for example, I am A positive, but my mother is not A positive, isn't it? My mother is having a different blood group. She is B positive. So if I am A positive, she might be, I'm not giving you an example of that, yes, but she is not A positive in any ways. So there is a difference in blood grouping system. So that is basically why, because I have evolved my own blood group, I am A positive, she is having something different, my father may be having something different, isn't it? So this all you are going to study in the next class, that is genetics, how this blood group system is controlled by genes. This all will be taught to you in the next year. But now it is very, very clear to, like, should be clear to one and all, that we are not getting the blood from our mother, everything from our mother directly. What we are having is, we are having the raw materials. Like, calcium will be provided to the child for bone formation, the fetus. The particular components of the, suppose if you want to talk about cell membranes. Cell membranes, the major component is plasma membrane, which is lipid. Ma'am was teaching you cell. So you know now that plasma membrane has a bilipid layer, isn't it? So cell membranes, they are lipid, so they need something which is lipid, more of lipid. The cells are forming, majority of our body is protein. So the child needs many protein things. So basically what is going on over here, the whole story is revolving around the raw materials. So raw materials are basically passed by placenta through the mechanism of diffusion. I have taught you what is diffusion. Diffusion is the movement of particles or the molecules from their higher concentration to their lower concentration. So placenta will be passing everything, including oxygen. And it will be removing also many things from the fetus into the mother, including carbon dioxide. Isn't it? So if the fetus is forming, it is also respiring, it's also having a heartbeat, it must be making carbon dioxide also. We have done this thing in breathing exchange of gases. The tissues, when they do metabolism, they make a huge amount of CO2. So that CO2 will be again through the placenta given into the mother's side. But yes, placenta will always prevent the direct mixing of the blood. The direct mixing of the blood will not take place. Why? Because there is a barrier in between fetus and mother. And what is the barrier? That barrier is placenta. So until and unless this placenta is present, there will be no problem. But during parturition is the process of giving birth. What happens is, let's see. During parturition, what happens is, what's the thing? During parturition, placenta, because yes, placenta needs to be broken, because until this placenta is going to break, the fetus or the child is not going to come out. So placenta breaks, and there is little, just make a note of little mixing of maternal and fetal blood. So a little bit of fetal blood is going to enter into mother's system. So maternal blood was Rh negative, fetal blood was Rh positive. Now, the mother's body comes to know what is Rh positive. See, she was Rh negative. Can you see over here? She was Rh positive, and the fetus was she was Rh negative and the fetus was Rh positive. Now mother's body comes to know what is Rh positive. So what is going to be the next step from the mother? It's going to be safe, the child is going to be safe, the child is going to have no problem. But yes, what is going to happen? Mother's body is exposed to Rh antigen. Okay, so mother's body is exposed to which antigen? Mother's body is exposed to Rh antigen. Now, her body has a reason to fight. Whenever there is a reason to fight, we will encourage the fighters. So if the mother, she comes to know what is Rh antigen, her body will start to prepare a fight against the Rh antigen. So what is going to happen? Next thing, mother's body starts to prepare anti-Rh antibodies. She starts to prepare. She cannot do anything now, but she starts to prepare. Since the exposure of Rh antigens is very little, this is not going to damage her system. So if it is like, if you're having the thought process that Rh negative is transfused blood with Rh positive, okay, if mother is Rh negative and she is getting transfusion of blood, this is not transfusion actually. This is a very little amount of blood that her body actually is exposed to. So we will not use the word transfusion, we will use the word exposure. So if you are exposed to a situation, your body will start preparing fight back things. But yes, the fight back will not be of that high level that this is going to be fatal for the mother. So this is very, very safe for both mother as well as child. So here only placenta breaks and there is little mixing of maternal and fetal blood. Here only the first fetus, obviously who is Rh positive, is safe. We need to mention this point. This is of importance, that the first fetus or the first child with Rh positive factor is safe. Mother is also safe. Mother has just done what? Mother has just prepared certain antibodies which are against the Rh factor given to her. Okay, just exposed to her. So if this is this big body, she is given this little amount of, just negligible amount of Rh factor, so that just she can prepare some antibodies. Which antibodies? Anti-Rh antibodies. Clear with this, everybody? The story is yet to take a turn. Okay, Tamil long, clear, children? Yes, Tamil long, are we clear? Tamil long, just let me know if there is any problem. Okay, now this was this whole story of first pregnancy. The same couple, they are having their second pregnancy. Okay, we'll just take this to the last page actually, because the situation is going to happen after this only, we can understand this thing. Okay. So again, what is going to happen? There is a second pregnancy for the mother. So again, same probability would be there. What? The fetus can be Rh negative or the fetus can be Rh positive. Okay? Equal probability of both. So if it's Rh negative, just like the other case we had just now, there will be no problem. Because mother is Rh negative, fetus is Rh positive, negative, there is no problem. But yes, this time if the fetus is Rh positive, there is some issue going to happen. Let's see what. Now this fetus, if it is Rh positive, now this time the story is somewhat Placenta cannot prevent, okay, placenta cannot prevent the entry of what? The entry of antibodies. Which antibodies? Anti-RH antibodies in the RH positive fetus. So now there's a reason to fight, and there are fighters also. And these fighters, they don't have any hesitation in crossing the placenta. So what is going to happen? This particular fighters, they are now able to cross the barrier and enter inside the RH positive fetus. So RH positive fetus is having RH factor where? RH positive fetus is having RH factor on this RBC. Remember, I made this in red, like this. And just let me draw some antibodies. So these are the antibodies. You will be reading these, studying all these in detail in next year. But still, this is what? This is anti-RH antibody. And what is this? This is RH factor. So what is it going to do now? It is going to perform clumping. What is clumping? It's the same thing what would happen in a normal case also. If somebody is RH negative, positive, how they will be feeling if the, so this is again one RBC, this is its RH, this is its RH, they will be clumped. Somewhat this kind of okay. So this is what is called as clumping. So this is what? This is the fetal RBC. This is called as clumping or agglutination. Now if this clumping happens, what would happen? The RBC will be blast. The RBC will start to blast. So this is erythroblastosis. RBC is what? RBC destroyed. Which RBC? RH positive fetal RBC. Destroyed. So if all the RBCs of the fetus are destroyed in this way, obviously the fetus will be born with anemia. We all know what is anemia, isn't it? With anemia and jaundice. And this condition is called as, so this particular condition of a fetus of undergoing all these situations and then born with anemia and jaundice is called as this is R is called as erythroblastosis foetalis. Clear? All of you. So let's once again see this whole situation, a bit tricky, but yes, it's interesting. So the first pregnancy of a couple, Rh positive and Rh negative. If the fetus is Rh negative, mother is also Rh negative, we don't have any problem. No issues are faced. Placenta, why? Because placenta forms a barrier in between and prevents the mixing of the two blood from the fetus to mother and mother to fetus. But yes, during parturition, the placenta breaks and there is little mixing of the fetal and the maternal blood. So the fetal blood is Rh positive, maternal is Rh negative. Now mother's body comes to know what is Rh antigen. Till now she did not have any ideas. This is just exposure, not transfusion. We are not transporting a bulk of blood into her body, but we are just giving her negligible shot that can be tolerated by anybody. So what we have is mother's body is having exposure to Rh antigen. So once exposure is done, I'm giving you the reason to fight. What you will have? You will have the fighters. So the fighters are ready. What are the fighters? The pink Rh, anti-Rh antibodies. So these anti-Rh antibodies, what are they going to do? They are going to play a role in the second pregnancy. They will be just stored in the body for a long time. We all know that our immunity is dependent on these antibodies because these can be stored for a long time. They have certain property called as memory. So these antibodies will remain in the system as such whenever the second pregnancy reported, and if again the fetus is Rh positive, they will come into action. What is happening? The placenta, yes, it cannot prevent the entry of anti-Rh antibodies. It used to prevent the entry of the blood. So blood cannot get exchanged. Okay. Excuse me. So we have what? We have now anti-Rh antibodies in the Rh fetus. So this Rh positive fetus is actually going to encounter a new series of problems, a new series of issues where anti-Rh antibody is now going to face Rh factor. This Rh factor is going to bind to the RBC in this way that I have shown in the diagram. And this RBC, fetal Rh positive RBC, is going to interact with each other, and yes, they are going to bring about agglutination, this particular clumping. When there is clumping means what? This kind of structure formation which is complex. This clumping takes place. This clumping is actually the reason where the Rh positive RBC destroyed and fetus will be born with what? With anemia and jaundice. And this whole condition, this whole scenario is called as erythroblastosis foetalis. This is actually very, very dangerous. It can be fatal also. Just let me write one more point. This condition, yes, I know this is a bit, like, flowchart is a bit tricky, but yes, you can do it, I know. This condition is called, can be fatal. So many times the fetus is not able to survive also. Okay. Yes, Shillong, Tamil Long. Are you okay, everybody? Are you okay? Fine? Yeah, children. Are you okay? Yes. Tell me. Are you okay? After this long story? Yes, Tamil Long. Did you go to sleep? Yes, yet listening to my lecture. Yes, Tamil Long. Fine? So is this clear? This is of importance. This question can come in exam. These kinds of questions are very important for long answer type question in your exam. So This is very, very easy, but yes, just I have explained it in a very, very flowchart-like manner. That's why it might be like you might be facing an issue with the length of the answer. But yes, if you just go and write a paragraph, it would be very, very easy for you. Yes, everybody is okay? Fine? Just give me a thumbs up, Shillong, if everybody is awake. Let me see. Okay, fine. So I guess everybody is awake. Okay, fine. Nice. Tamilong? Children, you are not replying. What happened? Are we connected? Okay. I guess, did you copy everything? Shillong, Tamilong? Did you copy each and every line? Yes. Copy it? Tamilong? Did you copy everything? Yes. Very good. Okay. Can this problem be sorted out? Since science and technology today is so advanced, can we sort it out? Can we sort this problem out now? Yes, what do you say? Can we sort out this problem now? Because we all know our technology is so ahead, medical science is so developed. This is a small case for medical science, isn't it? Can we sort this problem out? Can we? Just think about it. Take 2 minutes and think about it and let me know, can this problem be sorted out? Just have a small discussion and let me know if this problem can be sorted out. Yes. Yes, Shillong. What did you conclude? Did you Google something? Did you Google something? Yes. Just use your own intelligence. You people are smart enough. I know. You can do on your own also. Did you Google something? Okay. Let me know what is the answer, what do you think about? What answer you have thought? What is your perspective, how it can be treated? How it can prevent this situation? Yes. No issues, you can share with me, no problem. I'm here to guide you. Just let me know. Tamilong, where are you children? Yes, let me know, Tamilong, what is your perspective on this problem? What is your opinion? How can this be sorted? Fast. Shillong, tell me. Quickly type your response. Quickly. We don't have much time left for the class. Yes. Quickly tell me your response. What do you think? answers. Why are you taking so long? Tell me. No answers. Should I tell you? Yes. Shillong? Don't know. Why? See, what happened over here? Okay, let us have a look at this story. What happened over here? Mother's body is exposed to Rh antigen. Now, what if I destroy this Rh antigen? What if I target on these Rh antigens that have come into mother's circulation and somehow cross this process? What you have to do is that somehow stop this process as whole. Isn't it? There can be a system, there can be a solution to this problem if I am able to destroy this Rh antigen. If I am able to destroy this Rh antigen, my Rh antibodies, anti-Rh antibodies will not form themselves. That's because no reason to fight, no fighters. So what we are going to do over here is that just as the mother's body is exposed to Rh antigen, what we are going to do, we are just going to just remove all these things. Yes, this was just for your understanding. Okay. What we are going to do is that, but yes, this is important. I'm going to give something which is called as anti-Rh antibodies. These anti-Rh antibodies are different from these anti-Rh antibodies what mother is going to prepare, because this is having a lifelong time. This is going to extend to lifelong, because she will be having her own cells, and in her own cells they will be preparing what? They will be preparing antibodies. So these cells will be preserved for always in our system, if this is the case. But these are what? These are antibodies of very short life. So what you're going to do is that you're going to introduce into the mother, what? You're going to introduce in the mother certain antibodies which are having a very short lifespan. They will like immediately go and get cleared out of the system. Immediately cleared out. This is going to be a lifelong process. This is going to be staying. This is going to stay in the system for a very long time. So these anti-Rh antibodies will be sent or will be injected in the mother. This is said to be ROGHAM. RHOGHAM. Sorry, GM. ROGHAM is given, this is injection which is given to her immediately within 24 hours of delivery. So this needs to be given to the mother immediately, because once the Rh factor starts to get into system, mother will start to prepare her own antibody. What we need to give is, we need to give artificial antibodies, prepared antibodies with a very short life called as ROGHAM into her system within 24 hours. And what is this going to do? This is going to kill all the Rh containing RBC. So what is ROGHAM is going to do? This clears out Rh positive fetal RBC. So what you are going to do, you are not letting this step to happen. This step will not happen. This story is not going to take place. If the story is not going to take place, obviously this all flow chart will be useless. The next processes will be useless, and the second fetus will also be equally safe as the first was. Clear, everybody? So what you are going to do? You are going to clear out, you are going to remove what? Rh positive fetal RBC. So all that RBC which was from the fetus having Rh positive nature will be cleared from the system. Everybody clear? Just give me a thumbs up. Shillong, if you are clear with this. Everybody. So you are going to give what? You are going to give a shot of antibody having a very short lifespan. Mother will prepare antibodies, same antibodies, but that will be having a longer lifespan because she will be preparing it from her own cells. And in this case, you are just going to give ready-made antibodies. They will go into the system and then get cleared out also very easily. They will not stay in the mother's system. That's why I have written over there short life, and here lifelong. Everybody clear? Shillong, Tamilong? Okay, long story, isn't it? But yes, we have completed blood components. We have completed the blood group system. We have done a lot of syllabus today. We have covered a maximum part of this chapter. We are only left with heart. Okay, so when we'll be meeting in the next class, did you copy everything? First, let me know this thing. Have you copied everything? Shillong, Tamilong? Did you copy everything? Yes, children. I cannot see any thumbs up, nothing. Okay, copied everything. Okay, fine. Very good. So, understood? Everything is clear? Clear? Tamilong, Shillong, everything is clear? Nice. So, what we are going to do next is, we are going to start with in the next class with the topic heart, and we will continue this chapter exactly like next class it is going to be totally heart. Let's see after that what happens. So just go back home and read everything once, refer to your textbooks, whatever is written in your textbook. Let me know if anything is not clear. Okay, just please let me know. You can talk to me on the groups also, WhatsApp group also. If anything is not clear, just let me know in any possible ways. Okay? Fine? Okay. So, good night. Tamilong, Shillong. Okay, missing other children. Fine. See you soon. Okay, bye-bye, tata. Thank you so much.